Hello everyone. Today I am here with another biochemical test in clinical microbiology, the citrate utilization test. Learning objectives: know the principle behind the citrate utilization test, perform the test correctly, know the interpretation of the test result accurately, clinical importance of the test in microbiology laboratory, understand its role in laboratory diagnosis of infectious diseases. What is the principle behind the citrate utilization test? The test is based on the ability of bacteria to utilize sodium citrate as the sole source of carbon and ammonium phosphate as the sole source of nitrogen. Here, citrate permease enzyme present in bacteria metabolize sodium citrate and form alkaline metabolic products that leads to increased pH. Bromothymol blue changes the color of the medium to blue at pH 7.6. Media used in citrate utilization test. So two types of media are used. So first, Kojer's liquid citrate medium and second, Simmons citrate media. It is a modification of Kojer's medium. In most of the microbiology laboratory, Simmons citrate media is mainly used. This media contains two substrates like sodium citrate and ammonium dihydrogen phosphate. What is the basic difference between these two media? So, Simmons citrate media, it is a solid medium, but Kojer citrate medium is a liquid medium. So, Simmons citrate medium, as it is a solid medium, it mainly contains agar and bromothymol blue as the pH indicator. Positive test is indicated by growth and change in color of the medium. But Kojer citrate medium is a liquid medium, so it does not contain any agar or indicator. Positive test is indicated by presence of turbidity in the medium. Here the pH indicator is bromothymol blue. At pH 6.9 it is green in color, but at alkaline pH 7.6 it becomes deep blue color. Let us discuss some important points during preparation of media. Place 4 to 5 ml of media solution into two tubes. Make long slant, small butt. What is the reason behind it? To increase the surface area for better inoculation and better visual interpretation. Culture and broth suspension should not be used. Broth may contain sugars. It leads to false positive result. Let us discuss some important points to consider during inoculation. Light inoculum with straight wire loop is recommended for streaking. Inoculate the colony in serpentine or fishtail manner on slant only. Inoculation on slant helps in spreading the growth and better observation of the growth. Procedure of the citrate utilization test. Take the pear colony from nutrient agar plate, then inoculate the slant with light inoculum, then incubate the tube at 37 degrees centigrade for 24 to 48 hours, then observe the biochemical reaction. If growth appears on the slant and color of the medium turns to blue, then it shows positive citrate utilization test. But in case of no growth and no color change or the media remains green in color, then it indicates negative citrate utilization test. Citrate positive organisms are Klebsiella pneumoniae, Enterobacter cloacae, Stachytobacter purulenti, Serratia marcescens, Salmonella enterica, Providencia stuarti. Citrate negative organisms are Escherichia coli, Shigella dysentery, Yersinia pestis. What is the diagnostic importance of citrate utilization test? To differentiate closely related members of the family Enterobacteriaceae based on their ability to utilize citrate. Second, helps in culture reporting combined with colony morphology, gram staining and other biochemical tests. Citrate utilization test helps microbiologists to identify unknown bacterial isolates from clinical samples like urine, blood, sputum, pus, etc. Clinical importance of citrate utilization test. So first clinical condition is the pneumonia, especially in hospitalized or immunocompromised patients. Example, Klebsiella pneumoniae causes severe lobar pneumonia. Second, bloodstream infections. Blood culture isolates are differentiated by using various biochemical tests. Then gastrointestinal infections helps to differentiate enteric pathogens from normal intestinal flora. Example, differentiation of Escherichia coli from Enterobacter and Klebsiella species. Identification of nosocomial pathogens. Several citrate-positive bacteria, especially Klebsiella pneumoniae and Enterobacter cloacae, are important hospital-acquired pathogens. The test contributes to their accurate identification. Let us discuss some clinical scenarios so that you can correlate the test better. A 55 years old diabetic female presents with fever, dysuria and increased urinary frequency. Urine culture grows a lactose fermenting gram-negative bacillus on clade agar. The organism is indole negative, urease positive and citrate positive. In laboratory, clade agar mainly showed mucoid lactose fermenting colony, then indole test negative, urease hydrolysis test positive and citrate utilization test positive. So what is the most likely organism? Answer is Klebsiella pneumoniae. Second scenario, a newborn develops fever, lethargy and poor feeding. Blood culture yields a gram-negative bacillus that is citrate positive, motile and lactose fermenting. Lactose fermenting colony on McConkey plate, citrate utilization test is positive, hanging drop method showed motile bacteria. Which organism is suspected? Answer is Enterobacter cloacae. Third case, a hospitalized patient on a ventilator develops pneumonia. Sputum culture showed a mucoid lactose fermenting colony. Biochemical test reveals citrate positive and urease test positive results. What is the likely pathogen? Answer is Klebsiella pneumoniae. Clinical importance, citrate positivity supports the identification of Klebsiella species, an important cause of hospital-acquired infections. Precautions during citrate utilization test. Using heavy inoculum to streak the slant may give false positive result. Preformed organic compounds within the cell wall of dying bacteria may release carbon and nitrogen to produce false positive result. When inoculating a battery of biochemical test media with the same culture, the inoculating loop should be sterilized to the flame first. After sterilization, the loop is used for inoculation on slant. Remember, any carryover of glucose or protein onto citrate medium can give a false positive result. So, citrate utilization test should be performed first to prevent any carryover of glucose or protein. The reaction requires oxygen, so cap must be loosened during incubation. Thank you for watching the video. Don't forget to share, like, subscribe the channel for more updates. See you all in the next session.